Extremophiles are fascinating organisms that thrive in extreme environments where most life forms would perish. The term extremophile is derived from the Latin words extremis, meaning extreme, and philos, meaning loving. These remarkable organisms have evolved unique adaptations to survive and even thrive in conditions that are hostile to most life on Earth. Extremophiles can be found in a variety of extreme environments, including hot springs, deep-sea hydrothermal vents, polar regions, acidic lakes, and salt flats. One of the most well-known extremophiles is the bacterium Thermus aquaticus, which thrives in hot springs with temperatures exceeding 70 degrees Celsius. Another example is the Archaeon Halobacterium salinarum, which can survive in environments with salt concentrations 10 times higher than seawater. Extremophiles are not only of scientific interest, but also have practical applications in biotechnology, such as the production of enzymes that function at high temperatures or in acidic conditions. Studying extremophiles can provide valuable insights into the limits of life on Earth and the potential for life in extreme environments beyond our planet. The discovery of extremophiles has expanded our understanding of the diversity of life and the incredible adaptability of living organisms. As we continue to explore and study these extraordinary creatures, we are sure to uncover even more secrets of their survival strategies and evolutionary history. Extremophiles are classified into several categories based on the type of extreme environment in which they thrive. These categories include thermophiles, psychrophiles, acidophiles, alkalophiles, halophiles, and barophiles. Thermophiles are extremophiles that thrive in high-temperature environments, often above 45 degrees Celsius. One of the most well-known thermophiles is Thermus aquaticus, which is used in the polymerase chain reaction PCR, due to its heat-stable DNA polymerase enzyme. Psychrophiles, on the other hand, are extremophiles that thrive in cold temperatures, often below 15 degrees Celsius. These organisms can be found in polar regions and deep-sea environments. An interesting fact about psychrophiles is that some can remain active and reproduce at temperatures as low as -20 degrees Celsius. Acidophiles are extremophiles that thrive in acidic environments with pH levels below 3. One example of an acidophile is the bacterium Acidithiobacillus ferrooxidans, which is used in biomining to extract metals from ores. Alkalophiles, on the contrary, thrive in alkaline environments with pH levels above 9. These extremophiles can be found in soda lakes and alkaline soils. Halophiles are extremophiles that thrive in high salinity environments, such as salt flats and salt mines. Some halophiles, like Halobacterium salinarum, have adapted to survive in environments with salt concentrations 10 times higher than seawater. Barophiles, also known as piezophiles, are extremophiles that thrive in high-pressure environments, such as the deep sea. These organisms have adapted to withstand pressures that are hundreds of times greater than atmospheric pressure. An interesting fact about barophiles is that they have unique adaptations in their cell membranes and proteins to maintain their structure under high pressure. The diversity of extremophiles in different extreme environments showcases the incredible adaptability of life on Earth and provides valuable insights into the limits of life and the potential for life beyond our planet. Adaptations to high temperatures are essential for extremophiles to thrive in environments where most life forms cannot survive. One of the key adaptations of thermophiles is the production of heat-stable enzymes that can function at temperatures above 65 degrees Celsius. These enzymes have evolved to maintain their structure and activity even in extreme heat, allowing thermophiles to carry out essential biological processes. An interesting example of a heat-stable enzyme is the DNA polymerase from Thermus aquaticus, known as Taq polymerase, which is widely used in molecular biology techniques like the polymerase chain reaction, PCR. Another adaptation of thermophiles is the presence of unique lipid membranes that are more stable at high temperatures. These membranes contain special fatty acids and lipids that help maintain membrane fluidity and integrity in hot environments. Additionally, thermophiles often have proteins with increased thermostability, allowing them to withstand the denaturing effects of high temperatures. Some thermophiles form protective structures like spores or cysts to survive extreme heat conditions. These structures help the organisms remain dormant until conditions become more favorable for growth and reproduction. Thermophiles have also developed efficient mechanisms to repair damaged DNA caused by heat-induced mutations. By studying the adaptations of thermophiles to high temperatures, scientists have gained valuable insights into the molecular mechanisms of heat resistance and stability. Understanding these adaptations not only sheds light on the limits of life on Earth, but also has practical applications in biotechnology, such as the development of heat-stable enzymes for industrial processes. The remarkable adaptations of thermophiles highlight the incredible diversity and resilience of life in extreme environments. Adaptations to low temperatures are crucial for extremophiles that thrive in cold environments, such as polar regions and deep-sea habitats. One of the key adaptations of psychrophiles, extremophiles that thrive in cold temperatures, is the production of antifreeze proteins. These proteins bind to ice crystals and prevent the formation of large ice structures that could damage cells. This allows psychrophiles to survive and remain active in freezing conditions. An interesting fact is that some psychrophiles can continue to grow and reproduce at temperatures as low as -20 degrees Celsius. Psychrophiles also have membranes rich in unsaturated fatty acids, which help maintain membrane fluidity in cold temperatures. These flexible membranes prevent the cell from becoming rigid and losing function in icy conditions. Additionally, psychrophiles often have enzymes with increased flexibility and activity at low temperatures. These enzymes have evolved to function optimally in cold environments, where typical enzymes would become sluggish or inactive. Some psychrophiles form biofilms or protective structures to shield themselves from freezing temperatures and harsh conditions. These structures provide a physical barrier against ice formation and help retain heat generated by metabolic processes. By studying the adaptations of psychrophiles to low temperatures, scientists gain insights into the molecular mechanisms of cold adaptation and the limits of life in extreme environments. Understanding these adaptations not only expands our knowledge of the diversity of life on Earth, but also has implications for astrobiology and the search for life in icy environments beyond our planet. Adaptations to high pressure are essential for extremophiles that inhabit deep-sea environments and other high-pressure habitats. These organisms, known as barophiles or piezophiles, have evolved unique strategies to thrive under pressures that are hundreds of times greater than atmospheric pressure. One of the key adaptations of barophiles is the production of pressure-stable enzymes that can maintain their structure and function under extreme pressure conditions. These enzymes have evolved to resist denaturation and maintain activity at high pressures, allowing barophiles to carry out essential metabolic processes. An interesting fact is that some barophiles have enzymes that are more stable under pressure than at normal atmospheric pressure. Barophiles also have membranes with increased rigidity and stability to withstand high pressures. These membranes contain special lipids and fatty acids that help maintain membrane integrity and prevent leakage of cellular components. Additionally, barophiles often have proteins with unique structural features that allow them to remain stable and functional under pressure. Some barophiles produce osmolytes, small molecules that help regulate cellular osmotic pressure and protect cells from the effects of high pressure. These osmolytes act as stabilizers for proteins and other cellular components, allowing barophiles to thrive in deep-sea environments where pressure is immense. By studying the adaptations of barophiles to high pressure, scientists gain insights into the molecular mechanisms of pressure tolerance and the limits of life in extreme environments. Understanding these adaptations not only expands our knowledge of the diversity of life on Earth, but also has implications for astrobiology and the search for life in high-pressure environments beyond our planet. Adaptations to high salinity are crucial for extremophiles that thrive in environments with elevated salt concentrations, such as salt flats, saline lakes, and hypersaline environments. One of the key adaptations of halophiles, extremophiles that thrive in high salinity conditions, is the accumulation of compatible solutes. These organic molecules help regulate osmotic pressure within the cell and prevent dehydration in salty environments. An interesting fact is that some halophiles can survive in salt concentrations 10 times higher than seawater. Halophiles also have specialized ion pumps that actively transport ions across the cell membrane to maintain ion balance and prevent salt toxicity. These pumps help regulate the influx and efflux of sodium, potassium, and chloride ions, which are essential for cellular functions. Additionally, halophiles often have enzymes and proteins with increased salt tolerance. These salt-tolerant enzymes have evolved to remain stable and active in the presence of high salt concentrations, allowing halophiles to carry out metabolic processes in saline environments. Some halophiles form protective structures like gas vesicles or biofilms to shield themselves from high salinity and osmotic stress. By studying the adaptations of halophiles to high salinity, scientists gain insights into the molecular mechanisms of salt tolerance and the limits of life in extreme environments. Understanding these adaptations not only expands our knowledge of the diversity of life on Earth, but also has implications for biotechnology and the search for life in salty environments beyond our planet. Extremophiles and their unique adaptations have significant applications in biotechnology, offering valuable tools and insights for various industries. One of the key applications of extremophiles in biotechnology is the production of extremozymes, enzymes that function optimally in extreme conditions. These extremozymes, derived from extremophiles like thermophiles and psychrophiles, have revolutionized industrial processes by enabling reactions at high or low temperatures that were previously unattainable. For example, heat-stable enzymes from thermophiles are used in the food industry for the production of dairy products, as well as in the biofuel industry for the conversion of biomass into biofuels. Similarly, cold-active enzymes from psychrophiles are employed in the detergent industry for efficient cleaning at low temperatures. Extremozymes have also found applications in molecular biology techniques, pharmaceuticals, and environmental remediation. Another fascinating application of extremophiles in biotechnology is the discovery of novel bioactive compounds with potential therapeutic properties. Extremophiles produce a wide range of secondary metabolites, such as antibiotics, antifungals, and anti-cancer agents, as part of their survival strategies in extreme environments. These bioactive compounds have attracted attention for drug discovery and development, offering new avenues for combating antibiotic-resistant pathogens and treating various diseases. Extremophiles have also been explored for their bioremediation capabilities, as some extremophiles can degrade pollutants and thrive in contaminated environments. By harnessing the metabolic diversity of extremophiles, biotechnologists are developing innovative bioremediation strategies to clean up oil spills, heavy metal contamination, and other environmental pollutants. Additionally, extremophiles are being studied for their potential in astrobiology and the search for life beyond Earth. The adaptations of extremophiles to extreme environments provide insights into the limits of life and the conditions that may support life on other planets. By studying extremophiles, scientists are expanding our understanding of the diversity of life and the possibilities for life in extreme conditions. The applications of extremophiles in biotechnology continue to inspire new discoveries and innovations across various fields, highlighting the remarkable potential of these extraordinary organisms. Future research on extremophiles holds immense potential for uncovering new insights into the limits of life and the possibilities for life beyond Earth. Scientists are increasingly focusing on exploring extreme environments on Earth to discover novel extremophiles with unique adaptations and bioactive compounds. By studying extremophiles in diverse habitats such as deep-sea hydrothermal vents, polar regions, and acidic lakes, researchers aim to unravel the genetic, metabolic, and physiological mechanisms that enable these organisms to thrive in extreme conditions. One exciting area of future research is the application of extremophiles in biotechnology, including the development of novel extremozymes for industrial processes and the discovery of bioactive compounds for pharmaceutical applications. Extremophiles are also being investigated for their potential in astrobiology, as their adaptations to extreme environments provide valuable insights into the conditions that may support life on other planets. Future discoveries in extremophile research may shed light on the origins of life, the evolution of microbial diversity, and the potential for habitable environments beyond Earth. With advances in genomic sequencing, metagenomics, and bioinformatics, scientists are poised to uncover new extremophile species, metabolic pathways, and genetic adaptations that could revolutionize our understanding of life's resilience and adaptability. The exploration of extremophiles offers a glimpse into the extraordinary diversity of life on Earth and the remarkable potential for life in extreme environments. As researchers continue to push the boundaries of extremophile research, we can anticipate groundbreaking discoveries that will shape our understanding of biology, biotechnology, and the search for life in the universe.